First of all, it's a great pleasure to be here one more time and to be able to talk to you about some of the work that we have been doing and celebrate this opening of this center, and happy to have been a part of the discussions that have led to this center for quite a long time. Now, the title that was given to me was this: ultra-high field MR at the border between physics and neuroscience, from human genome to the human connectome. You know, that I didn't choose that title. Yeah, he says I didn't answer his email. But I asked him, well, where did you get this title anyway? You know, I said, well, you know, I took it from another lecture you are giving, and that this is the title of the lecture. In June, you are giving, and I was thinking, which lecture is that? I don't remember giving a title like this. But then I remembered, I give a talk in Berlin in June, and Thoralf Niendorf, I don't know if he's here, but he's supposed to be here, I think. But Thoralf Niendorf, who organizes that conference, never asks for a title. He just gives you a title. And I thought it must be Thoralf who gave that title. And I looked at the Berlin program, and in fact, it is that title. So I am going to try to live up to that, and maybe a little bit superficially in this talk. Therefore, I don't have a lot of time. But I am going to actually talk about some physics, some and then connectome and genome at the end as well. However, I really thought the better title would have been, if you had given me some more time to think about this, not you know, physics, the neuroscience, and all of that. It would have been this title, which is Limoncello to columns and layers and ultra-high fields. Now, many of you may not understand why this is an important title, but Rainer is already laughing because he knows that, of course, I've known Rainer for quite a while, but our real collaboration started when one night in Sorrento, we drank a lot of limoncello. And and then every time we somehow celebrate centers and things like that, we always somehow remember this. And Rainer, therefore, I actually went all the way to Italy before I came here to bring you a limoncello. It will come. So, okay, well, let's, and unfortunately, it's not cold, so I was hoping that maybe I can have it cold so that we can toast it just after my lecture, for example, but it's not cold, so we'll have to wait for that. So let's go back to physics now that we have the limoncello under our belt. And what can I tell you about physics? I mean, you know, this field is actually is a triumph of physics and neuroscience. And you know that we all are aware of the fact that, for example, the work that Paul Lauterbur and Peter Mansfield did is very important. And Paul is they received the Nobel Prize in 2003 for physiology or medicine. And Paul was the first one to propose imaging with magnetic resonance with using gradient fields to encode for space. And then all of the talks that you're going to hear today on tractography, functional imaging, rely on EPI, and EPI comes from Peter Mansfield. But of course, that was really, you know, in science, we build on the accomplishments of people that come before us and forge a road through the forest of knowledge. And we all know that we give credit to Edward Purcell and Felix Bloch for having discovered the nuclear magnetic resonance phenomena in condensed matter. And you know, in 1952, they received the Nobel Prize for that. But many of you in the NMR field may not know that the first real spin transition and an experiment, which was not essentially a condensed matter experiment like what Edward Purcell and Felix Bloch did, but comes from Isidore Rabi. And Isidore Rabi, it was a atomic beam experiment, actually. But he had this atomic beam here, if I can find my cursor. Yeah. And then the beam was going through this magnet, and he caused nuclear transitions. These were spin transitions that he induced by radio frequency waves. And then that spin transition led to the deflection of the wave. And this is really, and he got the Nobel Prize for 1944 for having done the first experiment actually showing that there is a resonance phenomena that associated with nuclei. Now, Isidore Rabi did this experiment at Columbia University, where I was a student, and he was a professor emeritus at that time when I was a PhD student there. And but it turned out, I found out about later, I didn't know it at the time, that we lived on the same street in Manhattan, near Columbia University, and he died of cancer, and at the end, he had to get an MR scanner, and he was, of course, he's reported to have said, it's amazing that, what I started, which is true, what he started at the end came to be an instrument with which we image today. So, especially, it's an important lesson, especially, I think, these days for our colleagues, especially in the United States, where the long-term investment of view of science is really rapidly disappearing to a certain extent. So from Isidore Rabi, and of course Isidore Rabi builds on other experiments like Stern-Gerlach and Pauli and et cetera, et cetera. You can go all the way back. We came to magnetic resonance with exquisite anatomical detail as shown over here. Now, today, of course, we're going to talk about functional imaging quite a bit, and that comes from the Bold effect, which is another phenomena which relies quite a bit on physics and the understanding of what is happening when you are actually, when you have compartments of magnetic material that is compartmentalized, in this case, in blood vessels, which is deoxyhemoglobin, and that was described by my colleague Seiji in Bell Labs, and that led to functional imaging. This is from our effort, Seiji and I and Yuta, who's my wife at the present time, and not at that time, but so where we demonstrated that you can actually see visual stimulation in the human brain, and at the same time there was this experiment from Ken Kwang from MGH. Oh, sorry, that was not, this is something wrong, left over from another slide. That's actually 1.5 Tesla from MGH. But, then we progressed again, very much relying on physics. In 1993, we also published a paper. This was the first modeling paper looking at trying to understand where this relaxation phenomena is coming from, what it does, how it can be, how can we understand this? So these, essentially, the curves over here demonstrate what is the contribution of blood vessels containing this deoxyhemoglobin to R2 star, which is 1/T2 star, to this phenomena that we are detecting when they have different susceptibility gradients across the blood vessels, which is encoded in this quantity in hertz here. You can think of this as a magnetic field going up, or you can also think of it for a given magnetic field, the deoxyhemoglobin levels actually increasing in the blood vessels. So there were a couple of very striking features here, that somehow the blood vessel diameter is important. There's some averaging taking place over here, but not over here when the blood vessel is small. And then this particular averaging, this is an averaging due to diffusion, then results in something like, if you look at spin echoes versus gradient echoes, there's a vessel dependence. Now, many years later, of course, there were many modeling work from many different laboratories, but this is maybe one of the last, never the last, I'm sure, modeling paper by our chairman, Kamil Ugurbil, when he was working with me at the Max Planck Institute, again showing that there's a strong magnetic field dependence to this effect. And then the magnetic field dependence, what it allows is, in fact, you can sort of distinguish between contributions coming from these small vessels versus large vessels over here. These are spin echoes, but in the paper there are a lot of simulations for gradient echoes. And in general, we don't like to see contributions coming from the large vessels, so we need to see small vessels. But it was really this work at the very, very beginning in 1993 when just shortly after, I mean, this modeling was being done when we were already conducting the functional imaging experiments. And it got us thinking, okay, how can we improve the specificity of these signals? How can we improve the functional imaging signals? I mean, they were very small. We couldn't even detect anything from my brain or Sage's brain, and Jutta was the only person that gave strong signals, and so we used her all the time as a subject. And how can we do that? And that led to this push to higher magnetic fields, as a result of which today this laboratory also has very high magnetic fields. The first thing was this 9.4 Tesla 31 centimeter Bohr animal system, from which you have seen results already, actually. These are the experiments that you have done when you were in Minneapolis using this particular scanner. We did a lot of experiments which I cannot go summarize in this very short period, especially given the fact that our chairman, I know, is extremely strict, and I have to be careful about my time. But, you know, for example, we were really concerned about the question of neurovascular coupling at that time. Do we have enough accuracy in the physiology of the brain to be able to detect those high spatial resolution structures? At that time also, you know, optical imaging people were sort of, you know, confusing the field a little bit. Grünwald and his colleagues came up with the idea that, no, you really cannot, blood flow is not regulated accurately enough. You cannot detect structures like columns with optical imaging, which was, you know, intrinsic signals, which was an analogy, in analogy to fMRI, and then they claimed that we should not be able to do this, this in fMRI, and they came up with this expression, you know, brain waters the entire garden for the sake of a thirsty flower. So it doesn't have this blood flow regulation accuracy. And one experiment we did in the cat cortex at that time is image orientation columns, but with CBF, not with BOLD, and show that, yes, I mean, when you actually show this, you know, grating of a particular orientation, blood flow increases in certain areas, but not every single area. And you get a map like this, which looks like an orientation domain, and then you get an orthogonal orientation, and this flow map here, in fact, reverses itself. So that, so the idea was, well, in that paper, what we came up with, you know, cerebral blood flow is regulated at the level of cortical columns, and CBF, and then we measured the point spread function for that. It was 470 micrometers under a single stimulus condition. That's an important point. It's a single stimulus condition. In other words, we are not like subtracting two different stimuli and taking out common contributions. And so our conclusion is, well, capillary level control of CBF must exist. This was questioned. And then, well, this particular concept is really cannot be true, but instead, you have a situation where the brain really waters the thirsty flower while it sprinkles generously around it. And hence, we should be able to do column-level imaging, and hence, you know, you will hear, in fact, from Rainer quite a bit about imaging at the level of columns. We did other experiments, like, for example, looking at layers, imaging vasculature and histology in the cat cortex, and showing that you know, you have specificity to layers. So this is looking at in the cat cortex again. Again, you have a confound with large blood vessels here in a gradient recall experiment. You have, however, at high fields, a contribution coming from here, and you can, you know, tilt your detection in favor of the middle layers using spin echo, something like that. So all of those experiments meant that we have to get rid of our 4 Tesla magnet. It became a sculpture in the garden. Instead, we went to 7 Tesla to push the resolution of the experiments that we are doing with functional imaging, because all of these studies conducted at 9.4 Tesla and modeling had indicated that we should be able to get to the level of columns. I won't speak about the columns. You know, Rainer is going to talk quite a bit about that. We have been doing a lot of work together with him. But of course, at 7 Tesla, it became possible to do what was really possible with optical imaging in an invasive fashion in animal models, we were able to demonstrate that you can do it in the human brain so that you can get column level resolution. Now, this was fantastic, but of course at some point the field has to move from demonstrating that there are columns, okay, or you can detect columns. It has to move from columns to behavior. I mean, they must be doing something there, and we have to go actually, instead of just showing, oh yeah, we can detect columns, oh, there are columns over here and there are columns over there, but we have to somehow incorporate that into our understanding of how the brain is working and how actually we can go from all the way from this kind of level of imaging to behavior. And again, I hope that Rainer will be talking about that. However, if you want to go in that direction, you really have to cover more than these very small volumes, and we were very much interested in looking at the whole brain at that very high resolution. It's possible these days, you know, when you have at 7 Tesla with a lot of developments, you know, for example, very good gradients and as well as parallel imaging, that has been a major development for high fields. You can do imaging like this. This is 0.75 millimeter isotropic EPI, almost column resolution, but covering the whole brain. The problem is it takes a little bit too long. For example, if you have head gradients, you can do this in about 6 seconds, and body gradients, 9 seconds. It's a little too long for functional imaging studies. So at that time when we were able to do these images, we were very much interested in pushing the speed of acquisition. And we did that by going to multiband. And these days, I don't necessarily have to explain this technique, because it's becoming very much mainstream. But essentially, you, instead of getting simple one slice at a time, one after another, you excite multiple slices, and you collect all those slices simultaneously, and then you actually use parallel imaging principles to unalias those slices and separate it from each other. And so you use this, and in 2008, we demonstrated for the first time in the brain, looking at, for example, you acquire 4 slices, you can unalias them like this, and then we were able to obtain fMRI studies like this, where we had 88 slices in 1.25 seconds with 16-fold acceleration, 2D acceleration. Now, these images come from, as I had mentioned, multiband or simultaneous multi-slice. It's really an EPI sequence. I won't go into too much detail here, but in the EPI sequence, essentially, you replace this one pulse here with this excitation pulse with a multiband pulse that excites simultaneously multiple slices, and the rest is kind of reconstruction. There has been an important contribution coming from MGH and others. Here, looking at so-called caipirinha method was developed here in Germany, and an implementation of that in EPI coming from Setzenpap from MGH, and added some more acquisition changes to the gradients, and of course a lot of work in reconstruction, which still continues, which still improves this to this day, has led into images like this, where when you collect a single slice versus 8 slices, you really cannot tell the difference. 12 slices looks very good, and then at some point, of course, it starts failing when you increase the number of slices. It will depend on the magnetic field, the coil density you have, etc. You can evaluate these quantitatively. For example, in this case, we are looking at how much of this intensity here leaking into these other slices in a 3-slice experiment. This was published relatively recently. The year is missing, but 2013. And you look at 4 slices, 8 slices, and at some point it starts, of course, failing. But even this 12 slice, if you consider what people do in parallel imaging, is actually quite acceptable. And with all of these developments today at 7 Tesla, we can go back and obtain EPI images like this, and even at 9.4 Tesla, this will be doable, and even to a certain degree better, because high fields are actually really better suited for for parallel imaging. Now, all of this came to play a very important role in the human connectome. And in the human connectome, this started when we were doing all of this for the 7 Tesla. The connectome project came into existence. And here we are talking about, of course, a macro connectome in the millimeter scale. And it's based on both diffusion-weighted imaging, tractography, we've heard about it before in the first speech, and then resting state fMRI to deduce functional connectivity. Since I have to cover a lot of topics here, from early physics to the genome, I really don't have don't have a lot of time to show you a lot of things. But for example, in the resting state fMRI, we are just acquiring one image after another, and we are looking at the temporal fluctuations, correlated temporal fluctuations in the time series, in the fMRI time series. There is no task involved, and these are the kind of parameters that we use for that acquisition in the human connectome project. It's 2 millimeter isotropic resolution, so we excite 8 slices at the same time, so it's a multi-band factor of 8. And then we can cover the whole brain in sub-seconds. So it's 0.7 seconds to cover the whole brain. And then we actually sample those fluctuations with this type of temporal resolution. Now, you know, the Human Connectome Project is actually a consortium. In our consortium, which is composed of Washington University, University of Minnesota, and Oxford predominantly, with a couple of other universities coming in for MEG acquisition, etc. So the analysis is the responsibility of Oxford, and Steve Smith, does quite a lot of this work with resting state networks. And when you acquire so fast and you sample this resting state fluctuations very rapidly, you end up with data that you can actually analyze with very high number of components in the ICA. So this is a 200-component ICA analysis of the data, which you can classify and hierarchically organize. And now the images look, in these resting state data, look very well defined and highly granulated. So here's an example of this from one of these ICA components. It looks very much like for example, a motor task, but it's an ICA component of this resting state data, so there is no tasking involved. We're just looking at brain fluctuations, but in the human connectome data, we also have tasks, and we can compare them, for example, in this case, to left-hand movement tasking, and they look really extremely similar, giving us confidence that we are actually seeing, in some cases, networks that are indeed corresponding to areas that are actually functionally related to each other. You can do the converse of this, pull out a ICA component that looks like the other hand, and now you look at right hand movement, again, extremely good correspondence. Steve has also been working on analyzing this data. Sort of, this is a network of networks. These are ICA components that he's getting, and then they're correlated with each other, and from this correlation, and you can actually acquire, deduce quite a bit of data. For example, we can tell monozygotic twins from dizygotic twins very easily. The data easily gives a really great correlation with IQ, and you can tell the difference between males and females very easily. And we're hoping, of course, that there's quite a bit of this kind of analysis will come out of that data. Now, the other component is this diffusion imaging, and here in diffusion imaging, there was another development that we had to undertake, which was to go for higher gradient sets, and you will hear from Larry, probably he will talk about data coming from MGH with even higher gradient sets than what we have, but we are working with 100 millitesla per meter as opposed to 40 millitesla per meter, and having those gradients in diffusion imaging allows you to actually collapse these gradients in terms of time, push the amplitude up, because what is important is the integral underneath these gradients, and then shorten this echo time so that we can actually reduce the signal decay during this echo time to improve signal to noise. And this has a major impact in SNR. So for example, here we have I mean, this is no diffusion weighting, and this is 3 different values of diffusion weighting, 2 millimeter resolution, a standard 3 Tesla instrument with 40 millitesla gradients, and when you go to 100 millitesla per millimeter gradients, we now are collecting data with 1.25 millimeter isotropic resolution. This is a nominal resolution, and even though the resolution is significantly better going from here to here, with this gradient set, we actually achieve much better signal-to-noise in diffusion-weighted imaging. This has major consequences. Just one example, 2 millimeter resolution, what most people do today, you are looking at trying to find 4 tracks that actually traverse this plane, and there are a lot of false positives. You see it in the hand and motor area, many false positives, but when you go to this human connectome data now acquired with 1.25 millimeter resolution and B values up to 3,000, you can actually see those tracks very nicely. I'll show you one more example. This is this is publicly available data. So if you see it in this area where we are expecting, for example, it is known that arcuate fasciculus is supposed to traverse from here, you can get those tracks which are connecting primarily the language areas. Yeah, you can construct those from that seeded area, the tracks connecting the various areas in the frontal cortex and in the parietal areas. And from the resting state fMRI, you can also pull those language areas. This is just resting state fMRI. It's not a task fMRI with a seed in the Broca's area and generating the resting state data, showing that there's actually a very good correspondence. I want to talk a little bit about some of the 7 Tesla work that is going on in the Human Connectome project. We are hoping that possibly there will be work at 10.5 Tesla within the Human Connectome project, but that depends on this magnet coming in line. And it may be interesting, for example, to look at the differences between 7 Tesla and 3 Tesla acquisitions in fMRI. We are somewhat better resolution. There were reasons why we didn't go to higher resolutions, because we wanted to keep this temporal resolution very high. And this gives actually ICA components that are at this lower or better resolution. You get more ICA components at 7 Tesla, and I think that in the fMRI world, I'm sure the 7 Tesla data will be quite a bit better than 3 Tesla data. That is totally anticipated. But what was much more interesting is actually looking at the diffusion imaging at 7 Tesla. 7 Tesla has challenges for diffusion imaging. We don't have as high gradients, and the T2 is shorter, but we ended up with parameters like 1 millimeter isotropic resolution. We are accelerating in both the slice direction and in the phase encode direction. And because of all this bargaining that goes on about who gets what acquisition on this data collection, it is very valuable time. We don't have as much time as we only collect 40 minutes of data at 7 Tesla as opposed to 60 minutes of data at the 3 Tesla. But here are, for example, what the images look like. This is 7 Tesla with 1 millimeter isotropic, and this is 3 Tesla with 1.25 millimeter isotropic for 2 B values, and we get better SNR at this higher resolution and much better definition. And we don't have that 3rd B value for a variety of reasons, including the fact that we didn't get the time for diffusion imaging in the allotted scanning protocol. Fractional anisotropy, again, looks, I have to say, I'm really pleasantly surprised about what you can do with 7 Tesla and diffusion imaging. I was not so optimistic that we could beat a 3 Tesla with a very high gradient strength. And you can get, and what really surprises me still is that even though we don't have the 3000 B value, we still get somewhat better results with 7 Tesla. For example, this is somatotopic organization. Again, looking at in this plane, 7 Tesla with fewer directions. We pulled that organization out actually, quite nicely, and not so nice at 3 Tesla. It's pretty good at 3 Tesla, but not so nice. And these are some tractography. to the auditory cortex, and what is interesting again, that 7 Tesla seems to give somewhat better results. And even though there are, when it comes to quantitative evaluation of multiple fiber crossing, we are failing. I have only a few slides left, Kamil, and I'm almost finished. But what would be really fantastic is really for diffusion imaging, for example, if we could have these high gradients at a 7 Tesla. We are handicapped because of the 70 millitesla per meter gradients, and still we are doing extremely well at 7 Tesla. If we could have 100 or 150 millitesla per meter gradient, then I think it will be a fantastic machine for diffusion imaging as well as fMRI. But 7 Tesla has problems, not that 3 Tesla doesn't. For example, we're missing some areas here. They are fully recovered here, but they are missing here. These are these very well-known B1 homogeneity problems. I hope you can see it on the screen here. It's very nice over here. And these are B1 inhomogeneity problems. It can be solved with parallel transmit. I won't go into in detail. For example, using a multi-channel transmit coil, or even really better coils, better coil designs exist. I don't have time to discuss it, but for example, you can, when you image those slices and map the B1, it's very non-uniform, and that gives this problem of signals dropping out in the diffusion or fMRI imaging. And by using parallel transmit pulse design, you can fix that, and you can also control SAR. But the problem exists at 3 Tesla. It's just that it's not so bad that you don't get away with it. This is actually an actual flip angle map at 3 Tesla. It's also very non-uniform. It's just that it's not as bad as 7 Tesla, so that you can actually work with that. You can sweep it under the rug, but you cannot do it at 7 Tesla. Finally, the genome. I took this example from my co-principal investigator, David Van Essen, on the Human Connectome project. And what he did was he looked at, this was just, of course, exploratory. It doesn't really, it's something to be hopeful about, not necessarily definitive at this point. But he went to the Allen Human Brain Atlas and was looking at a particular gene, in this case, that codes for this CART pre-pro peptide. And it turns out that here it's very high, and here its expression is very low. And he went then to the human connectome data. He said, well, is this correlated with functional connectivity? So he put a seed here, corresponding to that one, and looked at what was happening over here. So this is anti-correlated. So in this case, in fact, it looks like there's from the, the network or connections are going together with the CART pre-pro peptide expression. So you will be able to have this data set at this point in the millimeter or 2 millimeter spatial resolution for resting state fMRI, about a millimeter spatial resolution for diffusion imaging. And you can, in fact, look at relationship of connectivity and connectome with gene expression, because rather lower resolution data is being collected by other people. Finally, I want to, of course, acknowledge a lot of people from CMRR who had worked on all the technical development on the Connectome project. And over the years, actually, I've covered a lot of territory, people working on animal models and human fMRI experiments, many of whom have left CMRR. They run their own labs at this point. And of course, in the Human Connectome project, our Oxford colleagues and our WashU colleagues, Matt Glasser and David Van Essen, and David, of course, is the co-principal investigator of the Human Connectome project. With that, I will stop here, and thank you for your attention.